on.
Good afternoon. This is Miriam Dickinson. I'm a biostatistician at University of Colorado and have worked with Allie extensively over the past few years in a cohorts. And I'm going to talk a little bit about some of the methodologic challenges we had with this cluster randomized trial that she described earlier. So first of all, a little bit of background. What is a cluster randomized trial? There are some key features of a cluster randomized trial that distinguish it from a traditional randomized trial. And the primary one is that the unit of randomization is a cluster, not the individual. Clusters can be defined in various ways. They can be a medical practice, a clinic. They can also be a geographic unit, such as a community or a county, or an organization, for instance, a hospital or a school. And one of the features of that is that individuals are nested or clustered within the larger unit. And as a consequence, all individuals enrolled in the study from a particular cluster will be in the same study arm. So why would you want to choose a cluster randomized trial design? A primary reason has to do with the target of the intervention. So the question you might want to ask is, does your intervention target a larger unit, such as a clinic or a community, or some aspect of the environment rather than focusing specifically on the patient? Another thing that you want to ask yourself is whether contamination is a potential problem if you happen to go with a patient-level randomized trial. If there are patients from different study arms in the same setting, such as the same clinic, can they exchange information or somehow influence each other? Or perhaps they influence the clinician delivering the care. Other considerations to think about include the setting. Where will the study take place? And in particular is context of interest. And sometimes feasibility drives that decision. Sometimes you're in a situation where you just have to deliver a single intervention to the whole cluster rather than being able to randomize patients and provide more than one intervention in the same setting. So how do I conduct a cluster randomized trial? Well, in planning and getting started with this process, there are some common issues to think about. So the first is clustering itself. Individuals within clusters tend to be more similar to each other than members of other clusters. And you can think about lots of situations where this is the case. This is a violation of the independence assumption that is the background of many of the analyses that we do. And power and sample size, statistical analysis, all these things are affected by clustering. So bottom line, you generally have reduced power for the same number of individuals, and that's dependent on the level of similarity of individuals within clusters compared to individuals in other clusters. It's possibly going to involve greater cost, but again, it's just another consideration that you'll have to factor in, and always involves more complex analyses. Blinding is often not possible. Clusters can be very different from each other, so you could have a good bit of heterogeneity among the clusters. And then generally, the number of units or clusters to be randomized is much smaller than trials in which individual patients are randomized. So there's a high potential for a covariate imbalance between study arms. So just a quick introduction to power and sample size to cluster randomized trials. First, we have to know something about the interclass correlation coefficient. This is this measure of how similar patients or individuals within the same cluster are relative to patients or individuals in other clusters. So in a power analysis, some of the key steps are, first of all, decide on your primary outcome, and then obtain an estimate of the intraclass correlation coefficient, or ICC. And this can be based on the literature or actual data that you may have from prior studies. Next, calculate the variance inflation factor, and I'm not going to go into that. And then once you divide your actual sample size by the variance inflation factor, you come up with the effective sample size, which you can see in the column there. And you can use that in a traditional power analysis. So this is a simple approach. And what you see here is that increasing the number of patients per practice, say in a medical practice, has less impact on decreasing the effect size that you can detect as increasing the number of practices. Excuse me. Sorry. I think we're going to have to stop. Jordan. So it could happen again as I talk. Sometimes it just goes by the wayside. Hold on, let me get a big swig of water and then I'll take it from there.
Sounds good.
So the study that Allison Kemp described, can we drop that and start over? So the example here is this population-based versus practice-based reminder recall study that was described previously. And just to review, the purpose was to compare two approaches to increasing up-to-date immunization rates in 19 to 35-month-old children in Colorado. And one practice-based arm and one population-based arm. So early decisions were around the unit of randomization. We knew right away that individual randomization was not feasible and didn't fit the goals of the study. So we decided that county would have to be the cluster and the unit of randomization. Context, we did have some interest in rural versus urban, and so we wanted to be careful about that, and that would be important in our findings eventually. We also knew that we could obtain baseline data, which was necessary for some of the procedures that we needed to do to randomize. So that was important. And we also knew that we would be able to obtain immunization records for all children in the age range with records in the registry residing in the selected counties. So those were all important factors in the initial planning. Now, there were some implications of using county as a cluster. For the practice-based arm, all eligible practices in the intervention counties were invited to participate in training, thus eliminating potential selection bias where you only use practices that volunteer to be involved. But all were invited. However, practice participation was not a requirement. So it's still a population unit that we're looking at. We're looking at all children in the county that met criteria for inclusion. Individual affiliation with the practice was not required for the data to be included. Now, in the population-based arm, Again, all eligible children, regardless of practice affiliation or not, would be included. And then finally, the analysis would be of the population in those counties. We had pre-specified criteria. I won't go into them so much here, but we did stratify by rural and urban location. So one of the initial challenges that we had to address was covariate imbalance. These counties were very different. There was a lot of heterogeneity among the clusters, and stratification alone is not always sufficient to overcome this problem. Imbalance in clinical trials is not a new problem, and there were methods developed in as early as the 1960s to address the problem, but they were not extended to cluster randomized trials until the early 2000s. And even then, relatively few cluster randomized trials use the approaches. So the procedure that's outlined, we have to have baseline data on all the units of randomization. So in this case, it was county level data on all the variables that we wanted to include in this procedure for covariate constrained randomization. And then we define a balanced criteria as the sum of the squared differences between study groups on relevant standardized variables, and calculate this balance criterion, and generate all possible randomizations. So you have a balance criterion for every one of these possible randomizations. Then next step is to establish a criterion for the maximum allowable differences between the study arms, and define a set of optimal randomizations in which there's an acceptable amount of difference between the study arms. And from that set, you choose the final single randomization. So we first generated all possible randomizations. We standardized the randomization variables. We generated a file containing each randomization and calculated group means. We weighted the variables equally in this study and calculated the balance criterion for each possible randomization. Stratification variables such as rural urban can be included in the process by limiting the final possible randomizations to be considered to those that are balanced. So again, the variables that we used were total number of children, up-to-date rates at baseline, and some race, ethnicity variables, income variables, and then some variables around the types of providers. Once we got to this point, we examined the distribution of the balance criterion and set a value for defining the candidate set. Early work used the best 10%, but some later work has changed that. But in the early stages of using this procedure, the most studies were fairly restrictive. And one of the things that we did here to look at how well our procedure worked was to compare the differences in the means on the raw variables for all the randomizations in the optimal set versus others. And then finally, we chose one randomization from the optimal set as our final randomization and assigned counties to study arms. So what you can see here, this is information about means for both rural and urban counties for Colorado that were in this study, and they're highly variable. What I have here is mean, standard deviation, and minimum and maximum on each of these variables. This is a graph of the distribution of the balance criterion by optimal group versus this is the optimal group in red, and in blue we have the remaining randomizations. It's actually completely disjoint, but you can't quite see that from the graph. But what you can see, what is really important here, is that simple randomization could result in extremely unbalanced study arms in a trial of this sort. So these are the final, this is the final set of randomizations. And what you can see here is that in the optimal set, the differences, possible differences between the groups are really fairly small. It's fairly well balanced. In the remaining randomizations, there can be very large differences between the two study arms in the trial. So the worst randomization from our optimal set also gives fairly good balance in this situation. And you can see that the differences between the study arms, even in the worst randomization in that optimal set, are not so great. So any of those that you would choose would be okay for your trial. So now we have the final selected randomization. It's stratified by rural, urban, and you can see here that they are fairly well balanced. In particular, look at percent up to date at baseline, and it's fairly close between the study arms compared to what might have been in just a simple randomization. So the next challenge was establishing a cohort. The baseline cohort came from data obtained from the registry in June 2010. The follow-up cohort was obtained in December 2010 at the completion of the trial, and the final analytic database involved matching the baseline and follow-up records. But in this case, we had a pretty good match. It was 98.3%. Now, there are data and analytic challenges, obviously, in these cluster randomized trials. We usually analyze these using general linear mixed effects models with study arm, county baseline up-to-date rates, and rural urban location included as fixed effect, and county as a random effect. Now, and practice as a random effect. We ended up using practice rather than county, so we wanted to use site of last service as the main random effect in there, and children were assigned to this sort of cluster since it accounted for quite a bit more variance than using the entire county as a cluster. Now, what we found when we tried to do this had some practical issues, is that there were a number of children at every county that had no practice affiliation, or there were very small clusters, and we ended up aggregating those and creating an unaffiliated cluster within each county. And that solved some of the convergence problems that we initially had with analysis. There's more detail on this in the paper, but And then we also performed some secondary analyses within practice-based arm only. And we were also interested in rural or urban differences. So we looked at those as well. The results have already been presented, so I won't go back through those. But in conclusion, cluster randomized pragmatic trials present challenges. There are some interesting challenges, some which you can anticipate and some which you may not anticipate until you get into planning and carrying out the trial. But in most cases, you can find reasonable solutions to the study design and generally to the data and analytic challenges as well. And so in certain situations, it's really the right design to use for certain kinds of trials. And I would especially like to acknowledge our data analyst, Brenda Beatty, for her collaboration on this project. Thank you.